Okay, here we go. Okay, so hello everybody and welcome to the June webinar of the Applied Evolutionary Epistemology Lab. My name is Nathalie Gontier and I will be your host today. So we have 3 speakers today and I'm very honored about that. We have James Shapiro here from the University of Chicago, Benedikt Hallgrímsson from Calgary in Canada, and then Francesco Balzan from here in Lisbon, who Francesco is currently a member of the Applied Evolutionary Epistemology Lab. Okay, so we are going to start off with James Shapiro. And so to shortly introduce him, Professor James Shapiro is Professor Emeritus of Microbiology at the University of
Not
Chicago.
emeritus, not emeritus.
Non-emeritus, oh, not emeritus. So Professor of Microbiology at the University of Chicago at the Department of Biochemistry and Molecular Biology. And he is a student of Barbara McClintock, where he worked with her on mobile genetic elements, and also a collaborator of Francois Jacob at the Institut Pasteur in France. And then he has also been working with Jonathan Beckwith in Harvard Medical School. And so beyond that, he is also very active as one of the founding members of the Third Way of Evolution, which is an initiative of him and Denis Noble and Raju Pokotil. And in that regard, he is also very actively pleading for an extension of the modern synthesis. And more recently, even he has also started to apply evolutionary thinking into cancer research. And so without any further ado, I'm very happy and very honored to introduce to you James Shapiro. Please.
Thank you very much, Natalie. Let me share my screen. And what I want to talk about today is how it is that evolution is not accidental, but something which is evolved in all organisms. And we'll see if I have time to get through this all. By the way, I'm not for an extension of the modern synthesis. I think the modern synthesis is wrong in many ways, and you'll see why as we go through the talk. I wanted to point out first that all life adapts genomically to various ecological changes at different levels and on different time scales. So physiological changes occur to genome expression, and they're short-term, often daily changes within physiological limits, and the changes are not heritable. They're carried out by response networks in the cells and organisms that undergo them. Epigenetic changes affect cellular differentiation, developmental processes, and they occur episodically within the individual lifespan, and sometimes they're transmitted across generations. We're learning a great deal about that these days, but they don't affect the structure of the DNA in the genome, just the way it's packaged. Then there are programmed genomic changes, which occur to take care of various foreseeable problems that a cell or an organism may encounter. So there's telomere homeostasis, which is the continual addition of sequences to the ends of chromosomes by reverse transcription. Phase variation, which means changing from one regulatory state to another, very common in organisms which can live in the wild, but also can live as parasites within other organisms. Antigenic variation, which is a changing of the antigens on the surface of a cell to evade the immune system. There's sexual variation. A number of organisms can do sex change operations on themselves, and they do this by rearranging their DNA. And this is seen in yeast cells, for example, so that a haploid yeast cell can have some of its descendants change sex and can mate with other descendants to make a diploid yeast cell, which has far more robust survival. There's macronucleus development in ciliated protozoa following mating. There's chromatin diminution in plants and animals after mating and during the somatic development, some of the chromosomes are eliminated or parts of chromosomes are eliminated. And then, of course, we have adaptive immune systems where the DNA is rearranged in order to generate antibodies which can recognize a virtually unlimited number of antigens. So these changes occur from every cell or sexual cycle to opportunistic occurrence within a population's lifetime. And they tend to be recurring and reversible. Now, these temporary changes are distinguished from genomic changes which occur on an evolutionary time scale. And there are macroevolutionary genomic changes that generate taxonomic originations. That is, they create new species or new taxa in major kinds of emergencies. And those can be exposure to antibiotics, pesticides, or chemotherapy assaults in various organisms, or in cancer, in the case of chemotherapy, loss of the food source, mass extinctions. And these trigger massive genome changes. And one good place to read about them is Henry Hang's recent book from 2019 called Genome Chaos: Rethinking Genetics, Evolution, and Molecular Biology, Molecular Medicine, from Academic Press. Now, conventional evolutionary theory considers genomes as isolated, stable entities unique to each species and attributes hereditary variation to errors and accidents. And here's a quote from Jerry Coyne, from a recent blog of his, saying mutational change occurs by accident or as a byproduct of something else, like a gene being accidentally duplicated or the ingestion of DNA from another species. But those changes occur whether or not they'd be good. That is, increase the reproductive output of individuals in the species that is mutated. Gene shuffling, gene duplication, horizontal gene transfer, all of these are biological accidents that are just the precursors of adaptive differentiation, the fuel of evolutionary change. And I would argue that that's incorrect because there are cases where we know of changes that are induced by various situations, such as antibiotics triggering plasmid transfer between bacteria, where the genome change is directly related to the challenge which has been presented to the organism. Now, living organisms avoid genome accidents in a variety of ways. So that what used to be thought of as accidents turn out not to be accidents. For example, errors in DNA replication are corrected by two proofreading mechanisms. There's a built-in proofreading in replicative DNA polymerases, which have an exonuclease subunit that will chew back a strand which is incorrectly incorporated a base, and then resynthesize it to prevent errors that occur accidentally during replication. And there's also post-replication mismatch repair systems where the cell can detect where there are mismatches in the newly replicated DNA and correct them by excising the incorrect nucleotide and replacing it with the correct one. There is base and nucleotide excision repair processes which work after mismatch repair detects mismatches. There's homologous recombination, where recombination can replace a damaged or incorrect sequence in the genome. If chromosome breaks occur, a relatively error-free process called non-homologous end joining can take place and join the broken ends back together. There are checkpoints to make sure that everything goes properly during the cell cycle, in particular during mitosis, to make sure that chromosome distribution is correct and the chromosomes are not left behind. And in all organisms, including bacteria, when all of these proofreading and correction mechanisms fail, there are programmed cell death routines called apoptosis in eukaryotes when part of a multicellular in cells that are part of a multicellular population. So a cell whose replication process has gone awry will sacrifice itself so that there are not mutated members of a population. And finally, there are systems for recognizing foreign DNA and cleaving that DNA and removing it from the cell. And those are restriction and modification kind of systems in bacteria and CRISPR-like systems in bacteria as well. Now, living organisms respond to genome damage or stress in a variety of ways which either do or do not induce genomic change. So there's non-mutagenic, error-free repair systems. And these can involve intercellular genetic exchange and homologous replacement. For example, in UV irradiated archaea, the cells mate with other cells and the damaged DNA is replaced by intercellular exchange. There are also inducible systems for base and nucleotide excision repair to remove damaged DNA from the genome. But there's also mutagenic, or what used to be called error-prone repair. And this is interesting because this is actually responsible for the vast majority of genome changes that were formerly thought to be spontaneous accidents. So there are non-proofreading translesion mutator polymerases, which do not do proofreading and which can induce mutations in the DNA. And they are inducible by DNA damage. And in E. coli, for example, the DNA polymerase 4, encoded by the DIN-B locus, is activated as part of the SOS system. And if you remove DIN-B, you knock down what are called spontaneous accidental mutations by a couple orders of magnitude. And in eukaryotic cells, in plants and animals, and studied most intensively in mammals, of course, is a process of called alternative end joining, which is very different from non-homologous end joining. It involves the DNA polymerase theta and other polymerases, which are capable of microhomology mediated template switching. We'll see this in a minute, reverse transcription, and untemplated polymerization and chromosome restructuring. In other words, if there's enough stress on the genome and something has gone awry in genome duplication or transmission, a process can begin which is capable of making all kinds of changes and creating totally novel DNA sequences which have not previously existed, or in creating new DNA structures and fusion structures of pre-existing sequences. And then finally, genomes can be reorganized by a variety of what I call natural genetic engineering functions. These are a wide range of DNA cut and splice activities, which can restructure genomes by cutting the DNA and putting it back together in new combinations. There's activation of mobile DNA elements and latent endogenized viral genomes in damaged cells. And then there are the processes of chromoanagenesis. That's a new term which is made up to describe widespread genome change, including chromotrypsis, chromoplexy, and chromoanasynthesis, etc. And I'll show you chromotrypsis and chromoplexy shortly. And these are rapid, massive, multi-site chromosome restructuring that occur in plants and animals and humans. And we first recognized them by studying cancer cells. Once they were recognized in cancer cells, they were found to be active in germline genomes as well, and in other organisms besides humans and other mammals. So here's an example of chromoplexy from a study of 2011 called Massive Genomic Rearrangement Acquired in a Single Catastrophic Event During Cancer Development. And what's interesting about chromoplexy, which means chromosome weaving, is that it involves only a subset of the genome. Here, it involves only 5 out of the 23, the 46, excuse me, 5 out of the 23 haploid chromosomes in the human genome. There's 147 rearrangements in this particular tumor, a chordoma. And what you can see is there are multiple exchanges within chromosomes or between different chromosome pairs. And these rearrangements occur and give rise to a clonal population, indicating that they occurred pretty much simultaneously in the cell culture. And from other characteristics of this pattern of multi-site rapid genome changes, it appears that this occurs during the active transcriptional phase of the cell cycle. And these multiple sites are joined together. And here's just a couple other tumors which have different patterns of chromoplexy. Chromothripsis is rather different. You see some interchromosomal exchanges here, but in these cells, what you have is multiple exchanges within a single chromosome or chromosome arm, or sometimes a couple of chromosomes. So here there are 239 rearrangements of chromosome 15. And that's a colorectal cancer. And in a thyroid cancer, you have 77 rearrangements on the short arm of chromosome 9. Here we have 85 chromosome number variations of various loci on chromosome 8. And here we have 55 rearrangements on chromosome 5. And the thing about cancer is that we're observing these massive genome reorganizations in real time. Obviously, they have to occur within one lifetime. And as I said, the way the inheritance goes, it looks like they came in a single event. And actually, this can be studied experimentally as well. And it has recently, since they were first described in 2011. And we now know that chromothripsis, which are the changes that are localized to one or a small number of chromosomes, occurs when a part of the genome, a fragment of a chromosome is not distributed at the metaphase plate and becomes localized in a structure called a micronucleus. And that's shown here in red. The micronucleus, the red stains the histones. The green stain here is replicating DNA. And here we'll see, we're going to see this in time lapse in a second, but I want to show you the progression that goes on. You have a chromosome in a micronucleus. The rest of the nucleus here stained green is undergoing replication. And then there's condensation of those chromosomes. And the micronucleus chromosome replicates. And then as mitosis occurs and the two daughter cells divide, the chromosome in the micronucleus fragments into many pieces and then is distributed to the daughter cells unequally. And they stick those pieces back together, and that produces the pattern of chromothripsis. And let's just look at that in the time lapse again. Here you go, you see the mitosis, the fragmentation of the chromosome that was in the micronucleus, and then the distribution to the daughter cells. So we can see these events in real time. And what's interesting about these events is that they involve some very complex genome rearrangements. And in particular, it appears that they use a special DNA polymerase called polymerase theta, which is the backup for canonical non-homologous end joining in mammalian cells, and presumably in plant cells as well. And there are similar polymerases in other animals and in more primitive eukaryotes. And polymerase theta is capable of generating a whole number of genomic innovations during mutagenic chromosome repair. If you lack the canonical non-homologous end joining, which is basically an error-free repair pathway, polymerase theta becomes an essential protein. And that's because theta mediated end joining becomes the only way that broken chromosomes can be repaired and joined back together. And there's a series of references here up on the various ways that polymerase theta restructures the genome. So when the cells are deficient in homologous recombination in tumors, they depend upon polymerase theta for repair processes. They can't survive without it. Polymerase theta introduces what are called genomic scars into repair sites, which show that they can introduce new sequences into sites where they have not existed before. Polymerase theta is a terminal transferase, which means that it can polymerize without any template and make totally novel DNA sequences. The polymerase can modify the 3' terminal ends of both DNA and RNA and as terminal transferase activity, and that can then be incorporated in the genome. And the signatures of non-homologous and Pol theta mediated end joining have been seen in embryonic stem cells. And Pol theta uses microhomology, and microhomology can mean as much as one base pair to join together two different sequences. And that relates to another feature of polymerase theta, which is that it can jump or leap from template to template and make combinations of sequences from different templates. And finally, it's recently been reported that Pol theta is capable of carrying out reverse transcription so that RNA sequences can also be incorporated into Pol theta mediated genome repair. And here's a diagram of some of the activities of Pol theta. You have a single strand extension at a broken end. It can extend that end without a template. You can template back on the same sequence, so you get inverted repeat sequences formed. It can template on another single-stranded region in trans. It can extend after carrying out the templated, in an untemplated fashion after carrying this out. And then it can join together and complete stable end joining and gap filling. And this creates a huge variety of different sequences. And they've recently been described in this paper at the bottom here from Umbright et al. Last year, mechanisms generating cancer complexity from a single cell division error. They've looked at some of these changes and they've sequenced them. And what is shown here are the results of single cell sequencing of a cell which has undergone chromothripsis. And what you see is changes in copy number of the sequence of various sequences along the chromosome. And this is chromothripsis on chromosome 3 in the human genome. And here are 6 regions which are joined together on there. And where they're joined is indicated by the length of these bars. So this first red region is copied to a short sequence, and then the polymerase jumps to this black region and copies it for a way, and then jumps over here to the blue region and copies it for a way, and then jumps back to the black region, copies it for a way, jumps back to the red region, copies a segment, and so forth. And as you can see, you're creating a complex set of novel combinations of genome sequences, some of which are not templated and therefore have no existing complex in the genome. And sometimes you find these as a primary clone, which means that they occurred in a single cell division and gave rise to a clone of cells. And you see the same pattern of template switching, which I've just described. And here's a sequence from a renal carcinoma showing that this kind of template switching occurs in cancer cells as well. I think I'll skip over these, which were just there to show that even mycoplasmas, the cells with the smallest genomes, have specific genome change operators. And I come to my conclusions, which are that living organisms have three major goals: survival, growth, and reproduction. So they behave what's called nowadays teleonomically or teleologically. They have goals, and they are to survive, to grow, and to reproduce. They use genome change systems to overcome ecological challenges, as I've just described. And living organisms are episodically confronted by insurmountable ecological challenges. That occurs at the periods of mass extinction, for example. When confronted by an insurmountable challenge, living organisms already possess the natural genetic engineering operators necessary for rewriting their genomes in order to survive, grow, and reproduce as new life forms, that is, to evolve. In other words, evolution is built into the genomes of all living organisms, and they are able to confront what would otherwise be insurmountable changes by changing. And that doesn't mean that every cell is going to survive or produce a viable progeny, but it means that the capacity to make a major genome reorganization and create a new taxonomic group exists in the face of these major challenges to survival. So that's what I have to say. Thank you.
Thank you very much, James. Questions?
Did I do okay on time?
Yes, you did very well. We are only 27 minutes, so we have 13 minutes for questions. Also, I want to point out that Jim has said that he will not be able to be present for the breakout rooms. So if you have questions, please ask them now. And also, he is.
James, may I ask you a question?
Anton, yes, please.
James, for many years, I've waited person who will answer me this question. I heard about alternative splicing, and I read that human nature probably. Do you hear me?
I hear you, yes.
Yeah, that human nature is coded not by genes itself, but through the alternative splicing. And probably we should analyze evolution not on the level of genetic mutation, but on the level of alternative splicing, because many meanings are coded during this alternative splicing. What do you think? Adaptation happens through the genetic re-engineering or through alternative splicing? Maybe we should search there, not through the gene changes.
Well, I'm not talking about gene changes.
Is it a clear question?
Yeah. Let me explain.
Okay. Yeah.
The modern synthesis was based on the idea that there are sets of genes, and then each gene encoded a particular phenotype, and that through the reshuffling and changing of those genes by accident, gradually organisms would change and evolve. We now understand genome function in a much more sophisticated, much more complex way. We know that there are processes like alternative splicing, like epigenetic regulation, like reverse transcription, like protein modification and so forth, like response networks, which mean that the expression of the genome is a more complex system than was ever imagined when the modern synthesis was formulated. Now, genome structure is also an important part of that. We know that expression is regulated in part, for example, by physical associations between different regions of the genome. And if you change genome structure, you change those associations.
Yes.
Already in 1940, Richard Goldschmidt was telling us that changes in chromosome structure were important macroevolutionary changes because they could change the phenotype in ways that single gene mutations could not. So I think alternative splicing certainly is important. It's an important part of regulation. It can lead to genome changes because the alternatively spliced RNAs can be reverse transcribed and incorporated into the genome, and they have been. But the whole process is very, very complex, and I think we're only at the beginning of understanding it. One of the major problems with the modern synthesis was the idea of junk DNA, the idea that repetitive DNA was devoid of meaning because they didn't code for different proteins. And we now know that repetitive DNA does two things that are very important in the genome. One is it formats networks in the genome for expression, or sometimes for things like alternative splicing, so that complex phenotypes are executed properly. The other thing that repetitive DNA does is it encodes a large fraction of non-coding RNAs in the genome. And we now know that non-coding RNAs, which don't code for proteins, are very important for genome regulation, for regulating expression and other phenotypes. And we're just beginning to learn about that as well. So I think we have to take a much more humble approach to understanding the relationship between the genome and the phenotype. And I would be far from the first person to say we should not ascribe everything to changes in DNA, because there's epigenetics and there's other aspects of heredity which I think we don't yet understand. I hope that answers your question.
My point is, maybe you mentioned engineering that we can rearrange DNA, but we also could rearrange alternative splicing too. So we could rearrange not only different steps. Yes, we have different levels of rearrangements. We could rearrange DNA, we could rearrange alternative splicing, we could rearrange chromatins. So there are many levels or steps where we can rearrange.
Yeah, I think we need to look at the genome as a database, which has a highly formatted hierarchical structure, and it's very complex. And for example, splicing sites are part of the genome and the signals which control that. And then there are these non-coding RNAs which regulate splicing, and how they're expressed and what they do is part of the control of splicing. So wherever you look at it, you have a complicated, complex system to understand. And the structure of the genome, what it contains, is very important in determining that, but I wouldn't say it's the only determinant.
Yeah, I see. Thank you.
There's also a question of João Pinheiro.
Hi, thank you for the talk. I missed the very beginning of the talk, like maybe the first minute, so you might have answered this question, but all these strategies or mechanisms that you covered, like the repair strategy and so on, I'm guessing they're not really ubiquitous to all living organisms, but throughout your slides, you did say living organisms this, living organisms that. So I imagine maybe some are, right, yeah.
Yeah, the three slides I didn't show were to show that even the organisms with the smallest genomes, mycoplasms, use DNA rearrangements to adapt to different circumstances.
So all these strategies that you covered are actually shared by all living organisms, quite literally. Okay, that's interesting.
Yeah. They don't all carry out chemotrypsis or all have polymerase theta, but they have some ways of rearranging their DNA.
Right.
And it's not an accident or a response. It's a very basic quality because living organisms live in an unstable ecology. And they have to be able to change. Change is normal for life. Change is essential for life. And we have to recognize that. I think the old attitude was that change is an accident, change is a mistake. And you're lucky if the change is positive. But we know that change is triggered by crisis. And sometimes that's how organisms have survived many crises in the history of life. At least that's my opinion about it.
Well, the picture will be slightly different when it comes to the changes originating in cancers, I take it. But do you think there's still a role for, well, you know, with the adaptive changes, even when it comes to cancer-related mutations, do they affect the germline at all?
Well, we find similar mutations and similar massive changes in the germline. Okay. So once people identify these multi-site changes in cancer, they looked in the germlines and they discovered them there too. So that occurs, it occurs in the human germline. And the thing about cancer is a macroevolutionary disease. The cancer is evolving to be able to reproduce and survive better within its ecology, which is the body. The fact that it will ultimately destroy the body is not relevant to the cancer cells' attempts to be able to reproduce and proliferate to a greater extent. And I use cancer only to say that people might say, well, these kinds of genome changes are impossible, but they're not impossible because we see them in cancer. And when we find them in cancer, we then look and find them also in plants and animals. And I've given a few references to that in some of my recent papers on the cancer changes. So the ability to make these complex changes is present in all organisms. And cancer is only a case where we can see it more easily and have direct evidence that it can happen. And it doesn't take thousands of years or millions of years to happen. It can happen quite quickly. And one other point about cancer is that it turns out that these changes can happen in response to cancer therapy. So it's my opinion that the therapies which people use to treat cancer, using what they call maximum tolerable dose to knock off a cancer, to kill a cancer, often induces the transition of the cancer from what could be a chronic tolerable disease into a lethal metastatic tumor, which ultimately will kill the patient. And that we should become aware that the way we handle cancer can influence its development, and be very careful about not doing something which will convert the cancer into something far more lethal.
In that regard, Jim, also because we are putting this online, if there are people with cancer listening to this, how would you advise them to handle their treatment? Because I think perhaps some people might give up on treatment hearing what you just said. So how would you advise people with cancer in that regard?
Well, there are a few oncologists who are aware of this problem and are devising means to treat cancers by what they call adaptive chemotherapy, so that they treat with sublethal doses and they use different agents and they change treatment when they maybe are at the risk of inducing some of these deadly transitions. There's a fellow named Robert Gatenby, Gatenby, We had a symposium on cancer and evolution last October, and it's present online. So if you go to Cancer and Evolution Symposium and look that up, you'll see the presentations at the symposium online, and you can get an idea of the variety of ways that people are approaching some of these issues in cancer treatment. But I think that the recognition that we're not limited to the kind of accidental small step changes postulated in the modern synthesis, and the awareness that therapy can sometimes trigger lethal transitions in cancer will induce people to find more effective ways of dealing with cancer and converting it, as I said, from a lethal disease into a tolerable chronic disease and extend people's lifespans in that way, much as we've done with HIV, for example.
And maybe one more quick question. Yeah, I don't think there are more questions from the others, right? One more quick question also, because you said that also in the beginning in the distinction between the third way of evolution and the idea of the extended synthesis. You said that you are against the idea of an extended synthesis because you think that the modern synthesis is wrong. And here also in regard to this cancer research, you know, it is the modern synthesis itself that is like causing these ideas. So in that regard, how would you see a new biology?
Well, I think we've gone from beads on a string and gene-based inheritance to a very complex dynamic system and very interactive and capable of changing its own database, which is the genome, when it's appropriately stimulated or stressed or challenged. And I think we have to recognize that most characters in life have a complex determination. There are these what are called genome-wide association studies, which people have been applying to cancer and other diseases, trying to find single gene or single locus associations. Whereas the fact is that most characters, in fact, all characters indeed, are determined by many, many different parts of the genome. Many, many different functions encoded in the genome are involved. And they're governed by complex regulatory networks and sensory networks. And we need to appreciate that. So we have to have a more cybernetic, but I don't think cybernetic is an adequate word, view of how inheritance works, and understand that it also applies to the process of genome change.
Thank you very much, James. And so James Shapiro is not able to join us for the breakout rooms, but he has given me the permission to share his email address, and anybody, also the people listening to these talks beyond the recording on YouTube, everybody is very welcome to contact him. And so his email is sha@uchicago.edu. So edu from education. So thank you very much, James, for your very interesting talk. And so now we are going to move over to Benedikt Hallgrímsson. Benedikt Hallgrímsson is a scientific director at Basic Science at the Alberta Children's Hospital Research Institute, and the child health. He's also a child health and wellness researcher and in precision medicine and disease mechanisms. He's the head and professor of the Department of Cell Biology and Anatomy, and an adjunct professor of the Faculty of Veterinary Medicine, and an adjunct professor at the Department of Anthropology and Archaeology. He's also the editor-in-chief of Evolutionary Biology, and also very well known for a book that he did with Brian Hall on epigenetics. And so he's in Calgary, the University at Calgary, and I'm very happy to introduce to you Benedikt Hallgrímsson, please.
Okay, thank you very much, Natalie. So it's a pleasure to talk to this group today. It's a very interesting group, and I very much enjoyed the first lecture, James. And in some ways, even though what I'm talking about right now is very different and much less molecular, it follows nicely on what James was talking about, because I'll be talking about the importance of studying systems and the complexity of biological systems above the gene level. So the work that, let's see, my slides are not progressing. This works, okay. So the work that we do in our group, I often describe it as the developmental genetics of complex traits. And we do it really from two different but surprisingly mutually reinforcing perspectives. One is an evolutionary developmental biology one, that is to say, where we study how development structures the expression of phenotypic variation and how that relates to evolvability and to evolutionary patterns. And the second perspective is structural birth defects. So we study how variation produces anomalies that are classified as disease or dysfunctional, and we study the way in which developmental processes result in those in relation to the generation of normal variation. So the primary focus of much of our work over the past, I would say, 10 years or so has been the craniofacial complex, and both in a few model organisms, primarily mice, but also chicks, but also in humans. And so we've done a fair bit of work in humans over the past few years. And the human face and the faces of mice and chicks are in many ways classic complex traits. They are highly variable, highly heritable, and the variation is structured by development in interesting ways. And I think I'll hopefully convince you that we're getting somewhere in terms of understanding what we mean by that, the structuring of variation by development as we go through the talk. So as I said, the morphology of the craniofacial complex, and so including the human face, is highly heritable. We're all familiar with the fact that we look like our relatives, and so here are some examples of famous people, daughters and mothers and fathers and sons, where the similarity is quite obvious. And I'm sure we could find examples of pictures like this from our own family trees, where we see a striking resemblance between relatives. And this is a kind of an interesting thing, because despite the fact that within families, there are very strong patterns of inheritance. People will often talk about so-and-so inheriting their grandmother's chin or their grandfather's nose, etc. All of these traits are extraordinarily polygenic, highly heritable, but extraordinarily polygenic. And so genes that appear to have relatively strong effects within the limited context of a particular family, so responsible, for example, for the relatively discrete inheritance of a particular nose shape within the context of a family, those same genes have vanishingly small influences across the population. They're among thousands of other influences that are all interacting or adding up to produce continuous and extraordinarily polygenic variation in the shape of the face. And this is a phenomenon that we see a lot with complex traits. So here's just actually not a very up-to-date list of the genes that have been associated with craniofacial variation. A lot of money gets poured into mapping genes for complex traits, including the face. And the results of all of those analyses, and I have to admit I've participated and contributed to some of this work, but the results of it are really deeply unsatisfying, because you end up with very long lists of genes that then explain relatively small proportions of the total variance. I remember having a discussion with a colleague a few years ago, and who announced with great fanfare that the genetic basis for stature had now finally been resolved. And I don't think that is actually the case, but what he was referring to was that was this paper that came up with a list of 700 genes that explained 10% of the variance in stature, and estimated that there might be another 20,000 SNPs floating out there in the human population that explained the the sum total of variation in human stature. And I was struck by that, and I thought, well, what does it mean to say that 20,000 different SNPs might explain variation in stature? What is different from that than just saying that stature is heritable? There's no explanatory value really to the statement that something like a third or more of the genome might influence stature. This is the problem with complex traits. And it results from the fact that well, as I'll hopefully convince you, the developmental basis for complex traits is understandably complicated. But if you just think at a simple level of how genes actually influence morphological variation, you start to get a sense of this. And so one thing that one immediately needs to understand is that the effect of a variant of a gene on morphological variation is extraordinarily indirect. And so you have, for example, one could imagine sequence variation in a gene like FGF8, either in the gene or in a regulatory site that influences the expression of that gene. That then results in variation in the expression of the gene or potentially its function, which then affects some cellular process or even intracellular process. That then results in variation in some higher level process, some morphogenic process that is influenced by that cellular process, which in turn produces changes in tissues, and those in turn produce changes in the morphology of the organism that we're trying to study. And so we have many levels or processes through which genes are acting to produce variation. And that results in really complex patterns where you have phenomena like canalization and integration, two things that I'll talk about in this talk quite a bit. And you also have weird things like multimodal patterns of phenotypic expression within a genotype. This is surprisingly common. It's common, and you see it in human genetic disease. You also see it in the phenotypic spectrum of mutations in isogenic mice. This is a model that we're working with, for example. It's a mutation in the NOCID gene, which produces this extraordinary multimodal pattern of expression that ranges from holoprosencephaly to a variant of holoprosencephaly that has a fused maxillary process to a normal septation of the brain, but then a bifurcation of normal versus a cleft primary palate. All of these phenotypes can occur in individuals that have exactly the same genotype. So that's an extraordinary thing, but remarkably common. And so one of the questions that we are really interested in is, how does development generate variation like this? What is it about the above the gene level architecture of developmental systems that generates these kinds of patterns of expressivity and penetrance from mutations? So the short and simple answer, of course, is that complex traits are really complicated. And they're complicated in ways that are really only apparent when you study them in terms of their underlying developmental basis. This is if you look on this side of the slide here, you see Günther Wagner's very, very well-known depiction of the modular genotype-phenotype map, where you have genes influencing multiple traits, and of course, traits being influenced by multiple genes. So this is pleiotropy and epistasis in terms of connections between genes and traits. But of course, this is a vast oversimplification, because those gene effects are actually working through processes, each one of which could be seen as a complex trait. Each one of these processes is polygenic, and the processes are also interacting with themselves. And so they generate layers of complexity, which is ripe for producing all kinds of emergent phenomena as genetic variation gets translated through development to produce phenotypic variation. So Natalie asked me to talk about epigenetics, and I always feel a little bit sheepish doing that because I've used the term quite a bit, but I don't do epigenetics in the molecular modern sense. That is to say, very little of our work uses or is focused on methylation, acetylation, those kinds of molecular processes that have fall under the modern concept of epigenetics. We do epigenetics more in the Waddingtonian sense, and when we conceive of it that way, epigenetics really is it's a field that sort of started to come into existence in the wake of Waddington's work, and I think saw its heyday in the 70s, and then fell into decline in the 80s until virtually nobody uses the term epigenetics to talk about development in this way, and very few people actually have this perspective. And I think that's a shame, because regardless of what you call it, there is a need for a field, or for for questions to be addressed at the level of developmental systems above the gene level. Systems biology doesn't quite cut it, because so much of systems biology is about is essentially about omics and multi-omics integration. Waddington was talking about something a little bit different, and I'll come back to that. But when you think about this, what is interesting is that this field has the potential to bridge, I believe, the realm of quantitative genetics and the realm of developmental biology, because they actually address different questions, and that makes it difficult for these fields to talk to each other. Quantitative genetics identifies variants that contribute to phenotypic variation. Essentially, quantitative genetics is about variation. Developmental biology is about function and mechanisms, and not about variation. In fact, variation is a nuisance to most developmental biologists. Neither field specifically addresses the question of how genetic variants act on mechanisms to produce variation, to produce differences among individuals. That's the question that's missing, and the question that I think was had light shone on it by Waddington's work, but has now receded a bit beyond the lamppost in modern biology. So Waddington used the word epigenetics in two different ways, and that makes it, and I think one of the reasons that the use of the word has become confused since then is really this fact that he wasn't clear himself about what he meant by epigenetics, because he used the term to refer to two very different phenomena. So one is about, or one sense in which he used the word is about the minimization of variance in development. So the tendency for developmental systems to be buffered against both internal and external perturbations, and to produce consistent results. And so the famous bithorax paper, for example, is really about that phenomenon, about the minimization of variance, which interestingly has a epigenetic in the modern sense explanation, but of course Waddington didn't know that. The other sense in which he used epigenetics, though, is about is in the context of cell differentiation. So the idea was that that cells undergo branching patterns of differentiation, and the formation and the stabilization of those paths of differentiation was a form of canalization, in the sense that those paths could be viewed as creodes. These are completely different things. So the minimization of phenotypic variance and cells following bifurcating pathways, these are not concepts that should be linked by the same term. And I think the fact that he did that has caused much confusion in the field. And so when people talk about epigenetics, they often don't know, or often misunderstand each other, because they don't know the context in which that term is being used. So I'm going to talk about epigenetics in the former sense, so the sense in which variation is structured or limited by its passage through developmental systems. And we can think about that in two ways. So one is about how developmental processes integrate variation into patterns of covariation. So this is the concept of integration. And the second is how developmental processes modulate the extent of phenotypic variation that corresponds to genetic and environmental effects or perturbation. So this is the concept of robustness or canalization. So integration. It's probably familiar to most of you here, but integration is the tendency for traits to covary. Notably, integration is not covariation in and of itself. It's the tendency by developmental systems to produce covariation among traits. And so a great example of this, and I often use this when talking to clinicians, is a syndrome like achondroplasia, where there is a set of features that are correlated with each other, such as the protrusion of the midface, the frontal bossing, the altered cranial base angle. These are distinct aspects of the craniofacial features of achondroplasia, and they co-vary with each other, so such that individuals with more or less extreme achondroplasia have co-varying amounts of these features, and because they are linked by an underlying developmental process, which is a reduction in the growth of the chondrocranium, reduction of the growth of the cartilaginous growth centers in the skull, the synchondroses, which are the equivalents to the growth plates in the limbs. So, great example of integration and the clinically relevant one. So, the human face is highly integrated, or exhibits a lot of structure in terms of its variation pattern. And in that it's typical for many morphological traits. So just as an example, if we depict the human face with 5,000 different points, as we've done here, it can vary in 15,000 possible directions with those 5,000 points, because each one could vary in three directions. And of course, it's a bit artificial because adjacent points are not going to vary completely freely just by geometry. But it is still remarkable how much of the variation in facial shape is explained by relatively few dimensions. So here we're cycling through, I think it's 8 different dimensions of variation. It turns out that 15 dimensions out of these 15,000 possible explain the vast majority of the variation in the face. It's actually a bit lower than 99%. It's about, I think, 95% or something like that. But it means that what we think of as this remarkable diversity and complexity of facial shape variation in humans is actually a lot simpler than that, in that there are relatively few directions in which facial shape varies. Of course, those few directions are capable of producing the enormous diversity that we see, but that diversity is more highly structured than one would think. So just to give you an example of how this structure plays out. So here is so sexual dimorphism, for example, would be one of these directions of variation that accounts for a significant proportion of facial shape variation. About 6% of the total shape variation in the face is accounted for by differences between males and females. And here, one of my students, Jordan Bannister, has taken his own face and he's morphed it along the axis of variation that distinguishes males and females into a hyper female version of himself and a hyper male version of himself. This is all Jordan, but Jordan along this axis of variation between males and females. And interestingly, this same axis of variation also maps very closely onto the variation that syndromes that are associated with sex chromosomes. Also, they tend to fall along very similar directions. So Klinefelter syndrome, XXY, XYY, triple X. They tend to produce facial shape effects that fall along this line of sexual dimorphism. So that speaks to an underlying developmental a source of covariance that is then tapped into by a variety of different features. And interestingly, you could also map this, as one of my students is doing this, map this as a complex trait. So if you project the axis of sexual dimorphism onto the normal population, regardless of sex, you find out that it's a highly polygenic trait, which is interesting. So how does this integrated variation in morphology arise by development? So we've spent many years now, many different experiments, trying to figure out aspects of this. And this I'll just briefly talk about what I think of really as the first major one that we did. And this is work in collaboration with Ralph Mercutio and Nathan Young, where we used the chick model and manipulated sonic hedgehog expression in the face. Ralph, a few years before that, had discovered the frontal nasal ectodermal zone which expresses sonic hedgehog. It's a gene expression zone in the frontal nasal ectoderm that is set up by sonic hedgehog expression in the midline of the brain, and it influences the rate of cell proliferation in the mesenchyme that is sandwiched between the brain and the frontal nasal ectoderm. So what we found is that when we modulated the amount of sonic hedgehog in the face, you get this interesting nonlinear relationship between the amount of sonic hedgehog and the morphology of the face. And we've here quantified also the cell proliferation and the sonic hedgehog expression for these individuals. This is what that morphology looks like. You get a range of variation that goes from something that looks like cyclopia to mid-facial clefting, all driven by modulation of sonic hedgehog in the ectoderm of the face. So of course, this doesn't mean that sonic hedgehog is going to be a major explanatory factor for facial shape variation. In fact, when you look at the GWAS studies, which I've told you already reveal that facial shape is incredibly polygenic, sonic hedgehog doesn't really even come up. But it does mean that there is this underlying developmental mechanistic axis that is influenced by sonic hedgehog expression. You can manipulate it with sonic hedgehog expression that produces this axis of facial shape variation that maps onto normal facial shape variation. So it does mean that the regulation of mid-facial outgrowth via sonic hedgehog probably is an important determinant of facial shape variation, but not that genetic variation in sonic hedgehog itself is necessarily that important. So we wanted to explore this further because we had quantified in that first study the relationship between facial shape and the amount of sonic hedgehog expression. And one of the ideas that came up in the discussion surrounding that is, well, the sonic hedgehog expression domain, it has a particular morphology. What happens when you change that morphology? Do you also change the shape of the face? And Ralph came up with this beautiful experiment that we used to test this, where first of all, we had to develop a method for quantifying the shape of the fez, which we did in collaboration with Washington Mayo. But the experiment that Rolf came up with was to transplant brains from ducks into chicks and chicks into ducks. And the reason that might produce a change in the shape of the face is that it turns out that chicks and ducks have differently shaped faces at the time of the formation of the fez. And so they have a different morphology of gene expression. The zone that expresses sonic hedgehog is differently shaped in these two species. And so he hypothesized that if you transplant the brain of one species into the other, you'll get a transformation of the face in the direction of the donor species. Turned out that if you transplant the whole brain, the embryo dies, and so we ended up transplanting hemibrains. And so this is a chick embryo, and here's a duck embryo, and you can see the different morphology. And here's actually this is the correct label here. This is a transplant embryo, and so this is the chick side, which looks more like a chick, and this is the duck side, which, as you see, looks a bit more like a duck. And you'll see that the shape of the fez is also changed. It's changed in the direction of the duck, of the donor species, and this is generated by transplanting the half of the brain from a chick into from a duck into a chick. So you get a transformation of the shape of the chick face in the duck direction with this transplant experiment. And so we quantified this with morphometrics, and you can see here that both in terms of the shape of the fez, which is here, and in the shape of the head, there's a partial transformation of the shape of the transplanted embryos from the donor species to the recipient species. So what this means is it's not just what sonic hedgehog does that's important. It's not just how much of sonic hedgehog is expressed or when it's expressed. It's also important the morphology in which it's expressed. The three-dimensional spatial arrangement of sonic hedgehog expression is also an important determinant of facial shape variation. Okay. So following up on this sonic hedgehog work, where we modulated morphology by changing the amount of sonic hedgehog and also the morphology of sonic hedgehog expression, we decided to move this work into a genetic model. And after trying this with sonic hedgehog for some years, we abandoned that and and took up the FGF8 model. There was an existing allelic series that we were able to modify that had different levels of FGF8 expression. And so we created embryos with these different genotypes and quantified here the relative amount of FGF8 expression. You can see that we have this nice gradation from the wild type level down to basically nearly zero. And we hypothesized that this modulation of FGF8 expression would alter the shape of the face, because FGF8 is a really important gene in terms of setting up the development of the face. And that's exactly what we found. So we found that that modulating the amount of FGF8 expression produces a continuous integrated transformation of the shape of the face, which you can see here, from low FGF8 to high FGF8. You can see this is here in 10.5 day embryos, and this is in late fetal stage embryos. You see the same thing. But like with as with the sonic hedgehog study, what we found is that the relationship between FGF8 expression level and morphology was nonlinear. That is to say, there was a range from about the wild type level to something like 50% or 40% of the wild type level, where really nothing changed. That the morphology remained constant while FGF8 expression changed. So that's interesting. So you can tolerate a 50% loss in this key developmental gene. You don't actually change any morphology. But when you go below that threshold, then morphology starts to change very rapidly. And so you have this quick fall. And so as you go to the lower genotypes or the lower FGF8 genotypes, you get a very rapid change in morphology, both in the 10.5 day embryos and also here in the neonates. And we predicted from that and actually I'll come back to that later, I think, but we predicted from that you would modulate the phenotypic variance that you would see within each of these genotypes, depending on where you were on the curve. And so the basic model here is that if you are on a flat part of the curve, like up here, where an X amount of change in FGF8 expression actually produces very little change in phenotype, then because there's going to be some variation in the amount of FGF8 expression among embryos, that's going to produce a small amount of phenotypic variation. But if you're in the steep part of the curve, if you're down here or here, then the same amount of variation in FGF8 expression produces a large amount of variation in the phenotype. So this is a way in which nonlinear genotype-phenotype maps modulate the amount of variation that you get within a genotype. In other words, nonlinear genotype-phenotype maps are an explanation for canalization. So we dove into this a bit further to try to determine whether the underlying gene regulatory network that was affected by FGF8 expression, so the downstream genes of FGF8 gene expression, whether they were explaining any of this. So, for example, if you in the region of stasis, does modulating the amount of FGF8 expression not matter because there are other genes that then compensate, and maybe you run out of the ability of those genes to compensate when you fall below 50%? Turns out we didn't find any of that. In fact, the entire FGF pathway and its downstream the genes with the downstream effectors of that pathway exhibit this same nonlinearity. That is to say, the entire pathway appears to have a nonlinear genotype-phenotype map, and we don't really understand why that is. And we are in the process of designing a series of experiments actually to try to disentangle that. But it appears to be a fairly central architectural feature of this developmental system, that the gene expression to phenotype maps are highly nonlinear, not for all individual genes, but for the FGF8 pathway and its downstream effectors. Okay, so we've repeated this experiment in a number of different systems, and I'll just very quickly tell you about one. And I picked this example partly because it's recent, but also because it does involve a molecular epigenetic example. So this is variation in WNT9B expression in AWOLI-SNELL mice. These are mice that develop cleft lip and palate with incomplete penetrance. And the reason that they do so is that the Wnt9b strain has a mutation that is adjacent to a promoter site for Wnt9b. It's a retrotransposon insertion. And so it produces variable methylation of this promoter site, where embryos that are undermethylated will have reduced expression of Wnt9b. And so we quantified the underlying liability factor, which in this case was methylation of that promoter site, and constructed basically the same model where we predicted a nonlinear relationship between the amount of methylation and the phenotypic outcome, and showed that that was in fact the case. You can see that here, predicted the same outcome, and in fact you get exactly the same effect. So where are my variance plots? I should have reviewed these slides before I showed them. And anyway, you have to take my word for it. You get an increase in phenotypic variance and in the steep part of the curve, just as with the FGF8 mice. And the current focus of work in this area really is in the area of trying to figure out why you get these multimodal ranges of expression in models like the no-CYP null mice. And so we've at this point we've quantified this multimodal map using morphometrics. This is a recent analysis done by Marta Vidal in my lab that is very clever, because she was able to find a way to do continuous morphometrics from E10.5 right up to neonates. That's never been done before, because there's a lot of morphological change that occurs within this range, and quantified how you have different tracks of variation. So there's not continuous variation. There are actually 3 different tracks of variation that you see within these within the same genotype for these mice. And the model that we have that we are working on to explain this is that this is basically due to there being 2 levels of critical decision points, and that there is an underlying instability at the cellular level where decisions between where you're going, which track you're taking here, may be determined by relatively minor differences in the characteristics of the cell populations at that stage. And there's a critical cell population that we're focusing in, which is the central basal portion of the midbrain prior to the septation of the brain. And we're using single-cell RNA-seq to quantify the phenotypes at the cellular level of these different cells, and determining whether this model, whereby you have a variation in the gene expression phenotype of individual cells, determining whether individual embryos go one way or the other. So this is a way of connecting variation at the cellular level to variation at the organismal level. I don't have results to show. Well, we have some results in this, but nothing that really has come together in a coherent way. But we definitely have suggested evidence that this model is going to be correct. That these alternative tracks that are occurring within a single genotype can be viewed as alternative metastable trajectories, where the underlying metastability is the behavior of individual cells. And the direction that the entire embryo goes is determined by small differences in the population composition of the cells that are making those decisions. Okay, so how am I doing for time, Natalie? I'm worried I'm going a bit slow.
Yes, well, it would be good to wrap it up if you can.
Okay, so then that's why I asked the question. So I'm going to just skip over very quickly. The last point I wanted to make is, and I'll just make it very quickly, is that if variation is structured by developmental processes in the way that I've been arguing so far, one of the predictions from that is that mutations of major effect can recapitulate the background variance covariance structure. In other words, even though you have a trait like the face that is incredibly polygenic, determined by thousands of genes, you actually only need a few mutations to capture the possible directions of variation, because that variation is highly structured by underlying developmental processes. And we've looked at this in mice, and it's a bit of a complex story, so I'll skip over that. The simpler one is actually in humans. And so I'm in fact working on this paper right now. This is a morphometric analysis of syndromic variation in humans. It shows this is not individuals, these are syndromic effects. So this would be the average effect, say, of Apert syndrome, average effect of Sotos syndrome. And this is the morphology, the morphological variation in the faces that corresponds to these average effects. And it turns out that that a collection of about 120 different syndromic effects, in fact, recapitulates the background variance covariance structure for the human face. So this is important because it means that major mutations that are affecting the shape of the face are acting through the same developmental processes to produce the same covariance structure. They're often just doing it to a greater extent. So an individual, a syndrome like Sotos syndrome produces variation that doesn't go off in some different direction, that doesn't occur in the background population. It pushes variation along a direction that exists in the background population, but to a greater extent. It exceeds the range that you would see in normal variation. So syndromes push variation beyond the normal range, but they do so in exactly the same directions as other genes do, but just to a greater extent. And so this is a confirmation of this prediction that the variation is highly structured by developmental processes. Another prediction from this is that you can then take these syndromic effects, like the chondroplasia phenotype, or I talked about sexual dimorphism earlier, and you can treat them as complex traits and then map the genes associated with them. And it turns out that, for example, in the chondroplasia, a chondroplasia phenotype has a polygenic basis in individuals who don't have a chondroplasia. In other words, this set of transformations that characterizes a chondroplasia exists in the background population, determined by genes other than the ones that are associated with the chondroplasia syndrome. So this again is a confirmation of this general hypothesis that development, so that the morphology is structured by developmental processes. Those developmental processes can be perturbed in many ways, including by major mutations, but also by the conjoint effects of many smaller mutations. So they have a polygenic basis. And I think since I am out of time, I'll just say that the way to really get at this in the future is to start building conceptual tools and also technical tools to quantify variation, not just at the level of genes, but at the level at levels above the genes, from proteins to cells to tissues, and image them in 3 dimensions and in real time. And quantitatively relate them to each other. And this is the direction that we're pushing within our lab. We're developing methods not just to do morphology on embryos, et cetera, but to be able to integrate molecular information and morphological information in order to truly build an epigenetic view of embryonic development. Another aspect of this that is critical is to be able to work in highly standardized ways with very large samples. And so we're also pushing the frontiers of high throughput phenotyping, both in mice and also in humans. So building atlases and imaging and morphometrics pipelines to enable that work. And I would say just as an aside that as people build things like this, and we certainly believe in this, one of the really important things is that one doesn't build these things just for the purpose of your own question and your own lab, but you build them as community-wide platforms and share the data, because that greatly increases the impact of the work that we do. And so with that, I will wrap it up. Thank everyone in the lab who's done all this work and our collaborators, and I thank you very much for your attention.
Thank you very much, Benedikt. Questions? If you have questions, please. Is clapping. Okay.
Should I stop sharing?
Yes, please stop sharing. Yes, thank you very much. Questions. I have a question. When I hear you talk, and also when I hear James talk, like a very simple question, like, do you think it is knowledgeable like, you know, like the further we go, it becomes super complex. Like, you know, like
Mm.
is it knowledgeable? That is something that I ask myself when I hear like people like James and like you, Benedikt, talk about these things. Is it possible to know, to get to understand these things to the
So
fullest?
you didn't want to start with an easy question.
No, no, I did not.
So yes and no. The thing about biology, of course, is that it doesn't have to make sense. It wasn't created by somebody that had an underlying sense in mind, right? So it can be as complex um, as nature is capable of generating complexity, and that can be far beyond our ability to comprehend. So that's so the answer to that is possibly not. But in the case of the particular question that I'm asking, which is the structure of morphological variation, it turns out that for many things, like skeletal or facial morphology, the complexity is actually belied by structure. So the fact that developmental processes can be perturbed in many different ways to produce the same outcome is a really useful thing, because it means that while it may be impossible to understand the complexity of the face at the level of individual genes, and I actually think that's probably the case. And if you spend any time, say, reviewing NIH grants on craniofacial development, which I've done a lot of, you see a lot of the same story repeated for very different pathways and genes. And it starts to look like this really complex web that you can pull a string somewhere and generate a perturbation, and then you fix it, and you end up with a coherent story, but it doesn't add up. Like, it doesn't integrate with everything else to really tell you what is going on in terms of the important determinants of variation. You know, when, say, IFT88 and Wnt3 both produce similar effects and can be rescued, but they're completely disconnected from each other because the developmental mechanisms involved are completely different. Very difficult to build an understanding of development if you're focused on, say, the Wnt pathway or primary cilia. But if you are focused on the regularities of morphological variation and how they are determined by variation in developmental processes, then you can address your explanation at the appropriate level. I think we have a good understanding, for example, of how variation in the relative growth of cartilage and the relative growth of the brain produces variation in the shape of the skull and the shape of the face. Now, each of those things, the growth of the brain and the growth of cartilage, are really complicated. We understand a lot less about those things than we understand about how they integrate with each other to produce variation in the overall shape of the head. So the key here is to ask the question at the correct level to generate a tractable explanation. And that, I think, is what people don't do because there is an over tendency for to generate an explanation at the lowest level of reductionism possible. And that's not always the right strategy. Does that help?
It helps. It helps. Are there more questions?
I'd just like to make a comment, which is, I think we went through a period over the past century, perhaps, where we believed that reductionism was the way to answer questions in biology. And I think we're entering a period now where we realize that that's a fundamental error, and that the answer is at much higher levels of complex interactions and regulation and control. And we have to change our thinking so that we don't try and find the gene or the pathway which explains something. But we need to understand how all of these things work together to produce organisms that are functionally viable.
I completely agree with that statement.
In general, also, I think like these days, everything is very data driven. And in that regard, like before, there was a lot of theory like, you know, epigenetics, it's true. Like when you look at the term, like you go back to Waddington, which is like the 30s already, and that's ages ago in many ways. While today a lot of things are data driven, but then I feel that the theory is lacking behind. Like, you know, like very simple questions: where do genes, where does the genetic part stop and does epigenetics begin? Where does epigenetics stop and does development begin? Is such a difficult question these days, and there is not enough, I don't know, maybe translation of the data that is there to I don't know, maybe I'm like
No,
complaining.
I think you're right, and that may be reinforced in the way in which graduate students are trained in many programs. I think there is an emphasis on learning new techniques, and to be fair, there's a lot to learn if you want to be good in your field. You have to learn some really advanced techniques now to be successful, and so maybe that's all consuming. I know that when I was a grad student, so James, I actually did my piece at University of Chicago, where you are. I spent a lot of time with Lee Van Valen. I took evolutionary processes and evolutionary development from Lee. And the discussion around the table amongst the graduate students then was very different than the discussions you hear between graduate students now. Much more theoretical, much more conceptual. And I don't exactly know why that is, but I agree with you that there's there has been a move in that direction, and hopefully, if what James says is true, that we are moving in the direction of more systems level, more emergent property focused explanations, then I think the focus on the concepts may return. I certainly hope that's the case.
Would you like to comment, James?
Yes, I mean, I agree. And we're going through a period where it does seem that there's just a lot of data being collected. And the way I view that in genomics is simply that the data are telling us that the assumptions we had and the principles that were established some time ago are violated by the data. The conceptualization, I think, will come when there's a general consensus that the old conceptions need to be replaced, and we have to establish new ones. And so we're a little bit overwhelmed by data right now, but it is teaching us something important, which is that we can't rely upon the so-called verities of the past. And I think that's very appropriate for a seminar which is on philosophy of evolutionary processes.
Yeah, but there, I think then that's the problem. And like at this day, there are very few philosophers that can deal with the science, you know, because you have to have almost a double degree to be able to partake at the level that is decent to talk to scientists, you know, because otherwise, like, and there also, yeah, it's all about communication, education.
I agree.
Anton, yes.
Benedikt, I was surprised that in your conversation, yeah, you used the sonic hedgehog. I was curious who is the sonic hedgehog, but I found in Wikipedia that is just a protein, not an animal from cartoon. But still, may I ask maybe silly question? You mentioned this recapitulation theory, and I'm not sure. Did I understand you correctly? So what did you mean by this recapitulation theory ideas? For example, I thought before that, for example, lungs, they couldn't evolve just through the individual genetic mutation. We need a kind of massive mutation or mutation through the many generation. And you mentioned something in that phase also could change through the many generation? I didn't get.
No,
idea
I think you may have misunderstood what I said.
Yeah.
So
Yeah,
what I said was that you can get a dramatic transformation of the shape of the face by modulating one gene up and down by a large amount. And we showed this for sonic hedgehog and FGF8. What I was saying was that that doesn't mean that those genes are the ones responsible for the corresponding variation in natural populations. I don't think, for example, that FGF8 has much to do with the microevolvability of the face in mammalian populations, because the fact that it has this broad range of robusticity, where you change, where you are basically robust to 50% variation in expression on the downside. We don't actually know what happens on the upside. Means that you got to produce some pretty major changes in FGF8 to do anything, and then when you do things, you create big problems. And so I think the fitness landscape for genes like sonic hedgehog and FGF8 probably looks like a table, where there's a broad range of variation that is tolerated and doesn't produce any morphological consequence. But then you fall off that table and you produce very deleterious phenotypes. What that tells us about evolvability in a macroevolutionary sense, it's not quite clear actually. It probably means that a lot of the genetics of morphological change for features that people study, like shape of the face or something like that, are driven by genes that are peripheral to the central developmental systems. So the main pathways, the Wnt pathway, FGF, BMPs, sonic hedgehog, probably are doing very similar things across a broad range of species. And the morphological changes are driven by the secondary players or tertiary players that are a couple of orders removed from the central parts of the network. That's my intuition as to what's going on. That's a conjecture that will need to be tested. But I don't know that that's the case for sure. I think that's the source of what you were talking about. I wasn't talking about recapitulation per se. Does that help?
Yeah, maybe can we find kind of macro evolutionary trajectory for individual evolution or individual developments? Do you understand? So we can say that individual.
Anton.
Yeah.
Maybe we can take that to the breakout room. Yeah. Yes, because we're going a bit over time.
Okay, sorry.
Okay.
I'm gonna thank you very much. Thank you very much, Anton. Thank you very much, Benedikt. And so now we are moving on to the last speaker of the day, who is Francesco Balzan. Francesco Balzan has a background, he's still a student, he has a background in anthropology, and currently he's a master's student in philosophy at the University of Bologna, where he works with Raffaella Campaner. And so he's here with me now in Lisbon to read, because I'm giving him a lot of stuff to read. So he's very interested in the free energy principle as it was developed by Fritzson, and he's going to be talking about that today. And so the idea is to link that then to evolutionary epistemology, and also to learn about what we've been hearing a lot about, like systems theory and cybernetics, and how that works together. So Francesco, please go ahead.
Yeah, here I am. Many thanks, Natalie. Can you hear me and see the presentation?
Yes, we can see you and hear you.
Okay, perfect. So many thanks to Natalie, first of all, and to you all and the lab in general for this great opportunity. Many thanks to James and Benedikt in particular, even if this presentation is going to be for sure less molecular and rigorous in some ways, because you can situate it in the branch of philosophy of science. However, yes, it's probably more connected to this last meta discussion raised by Natalie about the deployment of complex or simple models to narrate science and to understand the complexity of nature in general. So as said by Natalie, the aim of my internship with her is to figure out the implication of the free energy principle for applied evolutionary epistemology. But however, as I know that the majority of you maybe is new to the topic, a huge part of the presentation is meant to unpack the free energy principle and then maybe introduce the most cutting edge implications and possible future paths. So let's start first by unpacking the free energy notion. So it is fair to say that the notion comes from thermodynamics and it stands for the amount of energy present in a system to do useful work. However, within FEP, the free energy principle will refer to a slightly different form of free energy, even if mathematically it's the same equation. That is variational free energy. That stands for an information theoretic construct, that means an approximation, that bounds, that means it's greater than, the surprise that may arise on sampling some data, given a generative model. So basically the variational free energy scores the probability of some data, given a generative model of how those data were generated. In other words, it is a measure of how well your model explains the external data. But what are those data and those models we're referring to? So let's unpack this notion with reference to the brain. That is actually where the frameworks originally originated and where everything always connects. So in the brain, the data are sensory inputs, obviously, and the model is considered as a statistical architecture encoded in the brain about how latent causes generate consequences. That is a probabilistic mapping, in other words, between the sensory inputs, the data, and its causes, so the possible source of this data. So here, free energy becomes a measure of the surprise that you would have in relation to data, given a specific generative model of how those data were generated. So basically, the free energy principle tells you that in order to exist, keeping the example of the brain, or to function in a proper adaptive way, you have to minimize this free energy that you can also call surprise or uncertainty, that is the contrary to model evidence. And in other words, you have to minimize the discrepancy between your internal generative model of reality and reality. So that means minimizing the discrepancy between how you expect the world to be and how it actually is. But why using an approximating measure? Basically, the free energy principle tells you that we use an approximating measure because surprise is intractable. So you can't compute it analytically, because this calculation would involve taking into consideration all the possible causes of your sensations, of your data. And that's why we constructed basically a quantity that can instead be evaluated. So before deepening into some of the basic aspects of these previous definitions, it's useful to understand the basic motivation of this framework. So it starts from trying to tackle this once very philosophical question, that is what it means to exist. What does it mean to exist? Self-organization here is surely a paramount feature. However, as you see in the left picture, in the world there are many self-organizing systems that are self-organized, but they self-organize to equilibrium, so they're not considered living systems. On the contrary, some systems exist in this world as living entities by self-organizing far from equilibrium, as this sea cow, a lovely one for me. In fact, she resists a natural tendency to disorder. That means she minimizes the entropy of her sensory states by basically limiting the set of states where she can find herself. So for example, she's very probable to find herself in water and not in land, as you, as I presume, are most probable to find yourself at a temperature of, let's say, 36 Celsius degrees. And to do this, you have to find a specific mechanics to avoid surprising states, like being out of the water for the sea cow, or being at 32 Celsius degrees for you, for example. So basically, the free energy principle provides us with this type of mechanics. So we have seen that unfortunately, we are unable to directly measure surprise, and this would mean calculating the probability of all the possible causes of your data. And that's why a variational free bound was originally constructed. So we have a pretty simple premise, no? But the problem is that this leads to complicated consequences, because the free energy principle claims that free energy minimization is what characterizes both macro and micro dynamics. So for example, natural selection is conceived as a way of minimizing free energy over evolutionary timescales through the increasing attuning of organisms with their environment. We'll see later that here natural selection is just one way of minimizing free energy by attuning the model to the world While, for example, through nature construction processes, and construction processes in general, systems change the word instead of changing the model to attune it to their internal model. So, and the same mechanics at the micro level takes place during perception, for example, in order to explain sensory stimuli. So as said by Carl Friston in this paper, the principle applies to any biological system that resists a tendency to disorder, from single cell organisms to social networks. Now, to give a bit more of context and situate this perspective, let's just glimpse on some fundamental authors and concepts. So here we have, for example, Ilya Prigogine and his focus from closed systems to open systems from equilibrium and the foundation of complexity sciences. We have autopoiesis from Maturana and Varela. We have the concept of homeostasis from Walter Cannon, foundation of cybernetics from Norbert Werner, and very importantly, Hermann von Helmholtz with his sign theory and the proposal of perception as unconscious inference. We'll also see that also this claim from Richard Feynman really cements in a way the epistemic authority of the fact. That is, if you can't build it, you don't understand it. So let's see a more familiar example of free energy minimizations. We've seen that the free energy principle casts the functioning of biological systems in terms of a single imperative, that is to minimize surprise. But how do living systems are able to minimize surprise or free energy in practice? So let's now imagine that I am a baboon and I glimpse a moving shadow in the bush. My sensory states now need to be resolved by identifying the most realistic source of that sensory information, no? So is that my teammate Charles, or is it a lion? That's a very relevant difference, as you can imagine, that needs to be solved in order to inform the choice of my next behavior, if I want it to be an adaptive one. In fact, if that shadow is Charlie, I can groom with him. If that's a lion, I should prepare myself for being chased, no? And crucially, the free energy principle here tells you that being able to minimize surprise in this situation to inform your next adaptive behavior is what really characterizes you as a living being. But again, how in practice? So we've seen that the baboon is facing the intractability problem, so he won't be able to directly calculate the probability of all the possible causes of that shadow. So basically, he ends up reducing an upper bound on surprise that we call variational free energy that measure the divergence between observed data and expected data. Importantly here, very importantly, the free energy principle states that these expectations about the sensations and their causes are encoded inside a generative model. That, in other words, is a probabilistic representation, internal, of the external environment encoded by the agent. Now, we obviously can't, we talk about probabilities and we can't talk about probabilities without mentioning Bayes. So here comes the Bayes. And basically what Bayes tells us here is that the first way in which a generative model enables the brain to reduce free energy, and that is pretty intuitive, is by changing its internal representations, no, of the external reality. That is, in Bayesian term, means moving a probability distribution from one prior belief, so before evidence, to a posterior belief, so after evidence. So back to folk psychology terms, it kind of makes sense not to think that sentient agents would like to carry around a model of the world, such that typically you look around and you say, yes, this is more or less what I would expect. So free energy basically scores the difference between what the system is expecting and what is actually seeing out there, but always in relation to an internal generative model. The Bayesian move, crucial here, is to cast this probability distribution encoded in the internal generative model that basically tells you how I expect the world to be in terms of Bayesian beliefs that must be updated based on evidence. Now perception becomes a form of Bayesian inference. So Bayesian perception, is the baboon's first way to avoid surprising exchanging with the world. And it is considered as a form of inference, as you can see. So you go from noisy data, that is the shadow in the bush, and you infer the most probable cause generated that generated the data, the lion or Charlie. And the probability of all of these different causes is defined in terms of a likelihood, that is the probability of that data given a specific generative model. So here perception is therefore a form of Bayesian inference, as it explains the data based on a probabilistic model of how those data were likely to be generated. And this likelihood is updated through evidence in a Bayesian manner, where the prior beliefs moves toward a more certain posterior beliefs by basically changing the internal structure of the model. That's basically learning, no? Intuitively. So just to recap again, Bayesian perception is therefore described as a form of Bayesian inference, where you change your internal representation about the external reality in order to attune your internal model to the external reality. So here you change the model based on external evidence, but this is again still half of the story, as we will see. So now we more or less have settled the basics of the argument, but let's now dive into a specific aspect of the free energy principle that is of crucial interest for me and my research, and hopefully for you too, that is the generative model aspect. So we referred extensively to generative models in general, but what is that? What is a generative model? So briefly recapitulating again, the free energy states that every NES system, that is non-equilibrium steady state system, performs Bayesian inference on its surrounding environment in order to maintain itself into a well-defined set of states. And this inference starts from the available data as observed states and find by inference the best causal explanation for the emergence of this data. The free energy principle tells you that systems do that crucially by leveraging a probabilistic generative model in the form of a statistical mapping between internal states of the system and external states of the environment. So now, as an historical context, it will be extremely useful to see how and why generative models were invented before being applied to the study of sentient systems. So here we go to Carl Friston. In fact, generative modeling techniques were invented by Friston in order to resolve a very technical problem related to brain imaging. That is to find the best possible explanation of the data produced by brain imaging, in this case, fMRI. So here explanation for Friston means to find the most likely brain configuration generated the data derived from fMRI. That is the bold signal, as you will see. So now keep in mind that via generative model, what we really want to do is to reverse this causality arrow that goes from left to right and move from the data, from the observed states, and infer the most probable, the most likely causal factor that generated the data. So in the specific case of fMRI, what stems out from the fMRI techniques is a bold signal. So bold stands basically for blood oxygenation level dependency and is basically a proxy of brain activity, as it is a sort of byproduct of neuronal activity consuming oxygen, etc. So this is a noisy data, you see, is like the shadow in the bush from the baboon. But this is the only data we have to explain a specific neurobiological activity. So what Carl Friston did with generative modeling is he constructed the most simple and still most explanatory model able to explain the bold signal data produced by fMRI. So this is the basic structure, as you can see here, of the circularity deployed by generative modeling. So on the right side, you have the experimental setup that generates the data, the bold signal. In this case, fMRI is the experimental setup, may be paired with, I don't know, specific task depending on the brain area you would like to ignite. And this experimental data from left to the center generates rough data, that is the bold signal. Now on the left side, you have constructed a computational setup in which a generative model provides you the most likely connectivity or causal structure able to explain your data. Obviously, the results provided by the generative model can in turn be used to refine the hypotheses and methodologies employed in the original experimental setup. So very importantly here, finding the most probable causal structure, the most probable neurobiological activity that generated your data, means finding the generative model that simulates the most likely causal structure producing your data. And under the free energy principle perspective, we've seen that this means choosing the model with the least variation of free energy. But how do we choose between them, between different models? So basically what the free energy principle tells us is that what you really want is a minimally complex and a maximally evident model able to explain your data. So this also means that what you want is not zero free energy or zero prediction error, as is defined with attention to the brain, because obviously a one-to-one scale map is completely useless, no? It overlaps with the territory, so it loses all the utility. So how do we quantify how well a model explains your data? We basically compare several models via Bayesian model comparison, in which each model encodes a different hypothesis of how the data, say the bold signal, were generated. And we compare them using an information theoretic construct that is variational free energy. So as seen before, variational free energy here is constructed as a measure of how well your model explained the available data. In other words, it tells you how much information is provided by every single model. So you can see there is a radical shift here proposed. So it's proposed that the radical shift proposed by the free energy principle is to deploy the above mentioned generative modeling techniques, not just to understand the data provided by an experimental setup, such as fMRI and the bulk signal, but to really understand the sensory signal of a living system. This is radically game-changing in that, and the radical game-changing move is that now this model has to involve the very same actions of the system in question. And crucially, this is because our own actions in the world are the main causal driver of our sensory data. Like here in the example, for example, in the picture on the left, you see how obviously eye sockets, that is eye movements or head and body movements, change the way you sense the world. No? This is pretty intuitive. And now these peculiar techniques deployed to construct models that are able to take into consideration the actions of the systems in the world is defined active inference. So active inference is a modeling technique that provides us a Bayesian mechanics to explain how action and perception are all in the same game of minimizing variational free energy. The system, in fact, deploys action and perception to gather evidence for the model that underwrites its own existence. And the action of the system is represented inside the model as its best way to minimize variational free energy. So basically here, we have added a peculiar move to this Bayesian picture. And that is, in addition to changing the model via Bayesian updates, as seen before, through perception and learning, variational free energy can be more directly minimized by changing the word in a way that will confirm our predictions encoded in the generative model. So here again, to clarify the role of action here, let's think back to our baboon facing a noisy signal. So we have seen that through perceptual inference, it will minimize free energy in a Bayesian fashion by rapidly changing its internal generative model as new evidence comes in. But action here is involved also at the perceptual level, as he will surely continually move his eyes and his body to gather new evidence for his hypothesis. For example, it is surely a lion. Or action is involved at a deeper level, as by just facing this uncertainty, he might just want to run away in order to gather evidence for a much more evolutionary fundamental hypothesis, namely that I am alive and that I want to continue to be so. And here it is. I know that's a chimpanzee, but I couldn't find a baboon smiling in the internet. I don't know why. So here we have made a further radical move, sorry. As we started from the fact that Bayesian model comparison between probabilistic generative models is scientists' best way to explain complex and noisy data, as we've seen with bulk signal with fMRI. So now what the FAB suggests is that, so okay, that's nice, but we are individual agents, we are scientists, and we are sentient, complex, adaptive systems, no? So maybe this is Bayesian strategy to explain data is not just a human prerogative, you know? So maybe this is also the best way for other complex adaptive systems to simplify the complexity of the world. So now Bayesian inference on generative models is intended as a mechanism deployed by systems to maintain themselves alive, rather than just a method deployed by scientists to describe sentient behaviors. And crucially, internal generative models such as live genetic model passed from being scientific constructs residing in scientists' brain to mechanistic models residing in actual life forms. So now it's fair to say that taking this revolutionary path, researchers started to apply this methodology to explain self-organizing behavior of complex systems around the world. And so we can see researches from molecular biology to sociocultural forms of self-organization. And we focus both on the microscale organization and on the right top, on knowing one place, up to the macro level evolutionary perspective, as in variational approach to nature construction. And we can also see that after the conceptual revolution of Bayesian inference and models as mechanisms deployed by systems rather than just descriptions contrasted by scientists, basically the free energy principle based inquiries have basically swum upstream. So up to the place where the framework originated, no? Basically the scientists constructing efficient models to explain the available noisy data. And this is exactly the aim of our research. So now the scientific method, as stated by my collaborator, John Campbell, is considered as a rediscovery of methods that nature has always used to perform knowledge accumulation. This methodology, as said before, being Bayesian inference to the best available model able to explain the data. And so science now, in our opinion, can be studied as a form of collective inference deployed by scientists to represent reality that with models that experience minimal surprise when encountering data concerning that reality. And therefore scientific theories maybe are basically second order models of models. And as such, and this is our central claim of the research, they might be further explained via third order model. That is a model of the scientific process that generated that scientific theory. So this is basically the major topic of my current research with some incredible Canadian collaborators. And as said in the beginning with Natalie, we're trying to articulate its connection with applied evolutionary epistemology by tackling some open questions. Some of them are stated here. So for example, can we really model science as a partially independent complex adaptive system? And what type of ontology does the peculiar epistemological move proposed by the FEP instantiate? And does the FEP framework applied to the scientific process legitimate a sort of superiority of science as an epistemic mechanism? And how are the different levels interconnected and how they influence each other? So how does causality flows from the different levels? How can the FEP in general inform applied evolutionary epistemology in its general agenda on how knowledge evolves and how it relates with the world? So I personally believe that by meeting the original aim that triggered the formation of evolutionary epistemology, the free energy principle seems a suitable framework for mutually informed classical evolutionary epistemology questions. And this research aims, for example, is to demonstrate in accordance with the applied evolutionary epistemology that knowledge in all its manifestations is a naturalized and evolving phenomenon. So that said, and in the hope that this presentation could have minimized some of your uncertainty about the free energy principle, I really, really thank you for your attention.
Thank you very much, Francisco. Questions?
Francisco, that was a fascinating presentation. It was a completely new topic for me, so learned a lot. It strikes me that there's an analogy to what is going on in the multi-omic world. In particular, the single cell transcriptomic revolution is confounding a lot of people, and primarily because the data are so noisy. And that's often viewed as a data issue, but I think it's actually a biological issue, that individual cells are much more variable in terms of their gene expression patterns than one would have thought, and certainly more variable than the processes that they sum up to create. Mm-hmm. And so I'm wondering if you see this same theoretical model being applicable in that domain.
Well, actually, I'm not so specialist on this topic. I take the philosophical aspect of all the framework, but as seen in the slide, there are many researches going on about morphogenesis and self-organization and how cells in a way communicate with each other. There's also a beautiful new article on cancer based on this free energy principle perspective. So constructing models based on the perspective that cells basically, they have an internal generative model of how the external reality is. And when this internal generative model is similar enough to the other cells, they basically, this is a way to see them interacting. So I don't know if the question is pretty specific. Sorry.
Yeah, go ahead. I wonder if there's conceptual work that is going on in parallel that is just not aware of this. So there's for example, one of the tools that's been developed for single cell RNA-seq analysis called SCPath, And a measure of gene expression variance that it outputs is called free entropy.
Mm-hmm.
So
I don't know anything.
remarkably similar.
Yeah, probably. I mean, this is more related to the meta-analysis of how you as a scientist construct models of the more complexity reality around you, how you get your data, and this is beautifully connected in a parallelism that if you have to bite the bullet in a way to see cells as working in the same way. Mm-hmm. And when you make this connection, everything gets really connected. Also this is in relation to applied evolutionary epistemology in general, mm-hmm. where knowledge is really flowing in through the same mechanics, that is Bayesian mechanics in a way. Mm-hmm. Yeah, I can suggest you some, I can share with you some bibliography in relation to,
that would be great.
To self-organization.
But one more question from a completely different perspective. So the principle of minimizing surprise, is that why dogs, for example, like things to be predictable even if they're bad?
Who? Sorry?
Dogs value predictability above everything else. So,
yeah,
if something happens that they expect to happen, they're happy even if that event actually is not good for them in any way.
I think that's a common trend between systems in general. But yeah, thank you.
I think Gerard also has a question, right? Gerard Jagers.
You're muted. You're muted, Gerard.
Okay, this is better?
Yes, now we can hear you.
So thank you, Natalie, for allowing me to ask a question, and
of course.
For the lecture or talk. My discipline is different than what you do, and therefore maybe that I'm a bit surprised that I see you using words in a way I'm not so used to. For example, free energy for me is something that means that you have energy that is still capable of performing action. Mm-hmm. So apparently in the work that you do, it has been redefined to something like a measure of surprise. Is that right?
Yes, this is right. I mean, I honestly, I don't know the mathematical technicalities, many of the mathematical technicalities of this concept. What I know is that the point of in common, apart that the equation is the same, you can use different interpretation of the same equation. And the most intuitive point of contact between the two concepts is that, as you said in thermodynamics, it is the amount of energy that is still usable for doing useful work.
Yeah.
And in this perspective, it is basically the divergence between your internal generative model and external reality, what you call the true posterior distribution. And this, in these terms, it becomes the amount of data or information that you can still grasp to really get a grip on reality. So it becomes the divergence that you can still minimize to get approximated to fit your internal model with the external reality. So I think this is a more intuitive point of contact between the thermodynamics term. So it remains an amount of energy, in this case is information, that you can still minimize, you can still use to get a better grip on reality.
Yeah. Do you now compare energy to information? Because information in itself, to me, doesn't really seem to have energetic value. It's just information. So.
Yeah, I see your point. I never wonder too much about these relations. I basically trust the colleagues that are working with me that deploys these mathematical equations to derive.
Okay.
And with different frameworks.
Yeah.
So yeah, I'm sorry I couldn't really deepen this topic.
It's just questions and
super useful.
Yeah. Okay. Another question would be if I am evolutionary, trying to survive or busy in an environment, and I am going to avoid all surprises. Where will that lead me?
What? Sorry, the last part.
When I'm going to try to avoid all surprises because I want predictability, because is it right that you say in your talk that systems are going for predictability?
Yeah.
I wonder where that would lead me in an evolutionary path, because if you never try new things, because you run in a circle of environments that you know, you will not easily discover something new, and maybe that is valuable for evolution, maybe.
Yeah, for
I
sure.
think the question that Gerard is trying to ask is, what
Yeah,
about creativity? Like, how does the free energy principle deal with creativity?
yeah, that's a wonderful question, and the topic is it's really no, it's really
It is a beautiful question.
it's a beautiful question and super interesting for the topic of energy in general. I didn't mention the technicalities of the difference between complexity and surprise and curiosity, but there are many researchers working on this, and there is also a problem raised by philosophers. That is the problem you Gerard are raising. So if this is the basic paradigm, why we don't sit in a dark room and we just close our eyes, and so we're able to predict everything. No, this is the basic argument we are raising. And there are many discussions on this, and what you a connection to this is that in the slide I show you that what you really don't want is a model that's really a one-to-one scale map. So you don't want to totally fit the external environment. So you want to leave this space for curiosity in a way. So you don't want to totally avoid external data, and you don't want to totally avoid prediction error or minimize totally free energy.
Just okay.
Anton, yes, you also have a question?
Just little comment, Gerald. If you are human or ape and you see a lion in the bushes, this surprise gives you energy. So information could give you energy. Just kidding. Okay.
Yeah.
Okay, maybe we can take it to the breakout room and then wrap up the official part of this webinar. Thank you very much, Francesco, for your talk. It was also his very, very first presentation, and I think he did it very, very well. Many congratulations. And thank you, everybody, thank you to the speakers for being here. Thank you to the audience, and also the audience that watches us after. And so the next webinar is going to be with Peter Gardenfors, Przemysław Zikwizinski, and Slavomir Wakiewicz from CLES, from the Center for Language Evolution Studies. And then that's going to be an entirely different topic. It's going to be on language, on learning, and on linguistic politeness. Okay, so thank you very much, everybody. Bye-bye.